not to be true. And one way that we know this is by the creation of this sheep called Dolly, which I'm sure you've all heard of before. And so Dolly is a mammal and she is cloned. So Dolly was created by taking a differentiated cell, putting it in the petri dish, and actually I have a couple slides on this. But we started with a differentiated cell, and we eventually were able to, from that single diploid cell, regrow or I don't want to say create exactly, but another organism was able to come from that diploid cell that was a legitimate organism with every single type of cell fate that there is, right? So it was a whole other new individual. So that could only have happened if the DNA from the starter cell was a full, complete set. Otherwise, we would have been missing critical pieces of genetic information. So this idea is called genomic equivalency, and it means that every single cell has the same DNA content. And I'm not talking here about, of course, cells might have small somatic mutations that happen in life or things like that, but in basic sense, the number of chromosomes, the length of the chromosomes, etc., is really the same from every single cell of our body. Okay. So that's genomic equivalency. So how was Dolly created? So you may, not sure if you realize this or not, but Dolly was created using a cell from a mammary gland, and so she was actually named after Dolly Parton. So this mammary gland cell was taken and put into culture and kind of grown for a while to let the cell settle down and de-differentiate a little bit. So lose some of those structural and physical characteristics of a mammary gland cell and go back to being a little bit more of a generic cell. Meanwhile, we need a donor egg. So what we have here is a different strain of sheep, have a black face, and we just took an ordinary haploid, non-fertilized egg and sucked out its genome. So you could do that by just putting a pipette in there and shoop, suck it out of there. And then we're going to now take this enucleated, so no more nucleus egg that has maybe any kind of special factors in there, or special environment, and fuse it with the nucleus of the mammary gland cell. So now we have actually a diploid cell. We have a diploid content of DNA within this cytoplasm that had originally come from the oocyte. So it's this sort of strange mixture of a new piece of DNA in a very embryonic or very early stage environment. And it will go on to divide and go through many mitoses. Now at some point, you're going to reach this stage called the blastocyst, where it's a multicellular organism, and it's done all of its early cleavages, and it's ready to now go through gastrulation and form a placenta. It cannot do that by itself in a petri dish, so it is surgically put back into the uterus of a mother sheep, and at that point formed a placenta just like normal, and kept on going and eventually became a baby sheep. Okay. So it's actually remarkable that this all worked, and in fact, it mostly didn't work. So the story behind the story is that they had to do this, about 434 times before they got one that finally actually made it through to become a functional individual. So, there's a certain cost here in terms of embryos and time and sort of like methodology that's not really a super robust way of doing it. It does teach us at least that biologically it's capable of being done. Okay, so getting back to the question of how do cells decide that they're going to differentiate to a particular cell fate, the more modern concept is that we do this by a process called differential gene expression. So gene expression refers to the part that each cell is turning on or off genes that cell needs at that particular time. Differential means that every cell does it a little bit differently. So we're all starting to do this in a slightly different fashion. So here is just a chromosome with 3 genes on it. In some cases, 2 cells express a certain gene in common. In other cases, they express unique genes. Okay, so which are the important genes for developmental biologists to study? We are much more interested in the genes that are unique and special to the cell as it's taking on its cell fate. And we're not so much interested in the everyday metabolic housekeeping genes. The term housekeeping gene means a gene that you basically need for the survival and metabolism of the cell. So something like RNA polymerase. Every cell has to have RNA polymerase or they won't be able to make any mRNAs. So that's not going to be that informative if you're trying to figure out, how did the cell get to be a muscle cell, because every cell does that. But on the other hand, if there's, say, some unique transcription factor that's turned on only in cells that become muscle cells, that's a good thing that we would want to study. A very informative gene that we could learn something about acquisition of muscle cell fate from that gene. So we love these specialty genes and we're less interested in housekeeping genes. Okay, the next few slides are kind of going to be a review of some of the things that you've seen in development in cell biology. And it's really nice to have had cell biology because you have now some of those principles in your mind about how genes are working and how they're turning on and off and different sorts of cell types. And we can bring those down to this class and think in sort of bigger picture terms about what cells are doing when and why and how they're getting together and achieving goals. Okay, so these slides are going to look at the idea of how is it that genes could be differently expressed from one cell to the next. And we start out here with the chromosome at the very top. It's going to go through transcription, processing, transport out of the nucleus into the cytoplasm. It joins up with ribosomes. It gets translated into a protein. The protein might get modified. And eventually you'll get some kind of an active protein after all of these steps. And really, any step along the way could be a way that one cell can do it differently than the neighbor cell. So it's not just whether we make an mRNA or don't make an mRNA. There are lots of opportunities here to have one cell become different than its neighbor. Maybe they do everything exactly the same way until they get down to this final step here where one cell adds carbohydrate, a different cell adds a different type of carbohydrate, and now we have 2 proteins that are functionally not the same. So there are many levels of differential gene expression. So how can we control what protein, final sort of functional item for the cell? How can we control when they're made? The first one is to look at the accessibility of the chromatin. So this is even taking a step back from gene transcription. We're just looking at how the chromatin, the DNA, is organized within the cell. And if some cells, for chromatin to actually be transcribed into mRNA, the DNA needs to be in a certain, more open configuration. And actually, let's look at this slide. So, as you may know, DNA is wrapped around these protein complexes. They're called nucleosomes. And if the nucleosomes in DNA are more spread apart, they're physically more spaced, it's easier for these big proteins like RNA polymerase to get in there and make mRNA. If they're very crunched together and tightly packed, then that's going to be generally associated with low transcription rates or none because it just can't get in there physically. So there are certain tags, such as modifications like methylation, that can be put on either the histones or the DNA that regulate how accessible the chromatin is here. So that's a method, or already a step of regulation. Hot area of research too, by the way. Things that are methylated tend to not be actively transcribed. So here are 2 different genes. Epsilon globin and gamma globin, they're right next to each other on the chromosome. One promoter is methylated, tightly packed in, the other is open, and one gene is being transcribed into mRNA and the other is not by this chromosome regulation. That's at 6 weeks. If we look at 12 weeks, now the situation is flipped because timing is important in development and everything is going to be different at a different stage. So what parts are methylated are now different. It can change. And what genes are being transcribed are different. So that obviously changes. Okay. We'll also hear a lot about transcription factors. These are really key proteins for developmental biology. And these are things that are going to bind to the DNA, typically as promoters or enhancers, and help decide whether or not that gene should be turned on or turned off. And so their two basic parts are a DNA binding part, which totally makes sense. Of course, they have to bind to DNA. But they may also interact with other proteins too in the cell so that you can get a very fine-tuned type of regulation just using these transcription factors. And keep in mind, we usually think of transcription factors as activators that would start up transcription, but equally well, they could be repressing it. They could be turning that gene off, which might be really important for that cell at that time. Okay, let's do one more slide and then we'll take a break. So these transcription factors in the cell, they combine to multiple different places. So here we have the coding region of gene X. This would be the place that we have the base pairs that would contribute to the amino acid sequence. Upstream of that we have a promoter, the TATA box, RNA polymerase is bound there, and a bunch of transcription factors. And then we have enhancers. So these are specialized regulatory sequences for that gene. And again, we often think of these as being upstream of the promoter, but they can be in different places. They could be downstream. They could be in introns. And amazingly enough, they could be 1 million base pairs away and still influence the transcription of that gene. So all of these little colored blobs, they're transcription factors. And you see there's a lot of them. And they all kind of act together to decide what's going to happen with the transcription level of this particular gene at this particular time. Okay, let's take a break. It's 8:50. Let's come back at 9, and we will resume on this slide.